Mentioned Dr. Paul Shadarevian, postdoctoral researcher working in Matthew Blurton Jones' lab at UC Irvine. He's working on the microglia replacement strategies, correct me if I'm wrong here, that are increasingly being considered for the treatment of primary microgliopathies such as ALSP.
Thank you. Can everyone hear me? I tend to also do a lot of interpretive dancing, so I'll try to keep my hands here the best that I can. Before I begin, I want to say thank you to the organizers. I want to say thank you to Glia Connect for inviting us, specifically Aaron, for really putting us and giving us this opportunity to interface with all of you, to learn from you as well, and to really have this inspiration that keeps us to do what we're trying to do within the lab. And as Dr. Spitali will mention a little bit later, this can be a very long journey, but we need this inspiration and we need this, patient focus to really think of, really creative and helpful ways to address not only ALSP, but also other neurodegenerative disorders. And so as I begin, I just want to highlight some of my, disclosures. Work, has gone into the development of, techniques that are now being used, by Savannah in order to really understand how we can take these approaches in order to help patients. And so I want to begin with this image. And I think this highlights really well how across a number of different neurodegenerative disorders, pathologies, misfolded proteins, these aggregates, they can occur within specific parts of the brain. They can occur throughout the brain and throughout the spinal cord as well. And so this has remained a really difficult strategy or limitation for us on how we can get good therapeutics into the brain. And there is this blood-brain barrier, this ability for the central nervous system to protect itself from unwanted pathogens and other infections that we can get in the periphery. But unfortunately, it's also limiting our ability to develop good things to the brain as well. And so this has really helped us focus on how we can treat the central nervous system from within the central nervous system. And so as Dr. Spitali also highlighted, there's been a lot of work that have really been focused on understanding what the immune cell of the brain is doing. And so microglia, they're always found at sort of the scene of the crime. Whether it's traumatic brain injury, whether it's any of these pathological proteins or these diseases we mentioned earlier, microglia are highly enriched, localized at these sites of infection, at these sites of injury. But they can also carry mutations that limit their ability to be protective, to be helpful, to do their natural job. And so this has inspired a lot of work on how we can target these cells specifically. And so I want to highlight some of those strategies and explain how people are going about this and maybe some of the limitations and challenges we continue to face. And so one of these approaches have been, well, why don't we use viral vectors? These are essentially small genetic syringes. They can deposit a gene directly into a cell. If we can engineer these cells to go after microglia, which are, they have a copy of a gene that's not producing or functioning the way it should, we can compensate for the loss of this genetic information within the DNA. And there's been some really great work showing within mice that we can develop these capsids and these really unique constructs. However, they're highly unreliable and very unreproducible. And I just want to highlight one example here. It might not be too clear, but this virus that was developed to target microglia with great specificity in someone else's lab was targeting the neurons and wasn't touching the microglia at all. And it continues to be a really, really difficult challenge to go after the microglia. Another approach is, well, why don't we use non-viral vectors? And this has gotten a lot of attention, especially in the treatment and the development of therapies for Alzheimer's disease. We can develop antibodies. We can try to engineer them to get into the brain. And then by having these antibodies bind to receptors that are not signaling as much as they should do, we can compensate once again for that loss of function within these these cells. And so there's been a lot of interesting work trying to go after, for example, TREM2. This is a receptor that's been shown in Alzheimer's to be really important to how microglia function. As microglia get closer to plaques, these receptors are upregulated. We see more of them on the cell surface. But again, although they've been really powerful within animal models, when we've translated this into the clinic, we've seen a lot of toxicity, especially within the periphery. And one of the reasons, too, that it's been quite ineffective within the brain is because although the surface receptor is expressed more and more, the internal proteins that are responsible for signaling into the cell are being expressed at a lower level as a consequence of that. And so this has also been shown in different animal models, too, that what was helpful necessarily for the plaques in the Alzheimer's disease models was now being detrimental for the tangles, the tau, the other pathology that shows up within patients with AD. And so we're helping one problem, but then we're causing a different problem within these animals. Another approach, well, why don't we deplete these cells? Microglia we know are very important, but we know they self-renew. And we know that as we age and our cells begin to age, they can begin to accumulate a lot of debris. They're getting stuck. They're unable to turn over. If we use these pharmacological drugs that can cross the blood-brain barrier, that can inhibit these receptors and essentially delete the microglia, we can then allow these cells to come back. They'll be rejuvenated and they'll be very young. And once again, in some studies, this is shown to be helpful in some cases, but it's proven to be quite difficult and quite detrimental, especially when we talk about demyelination. And what's really interesting too is in some animal models, this has actually exacerbated the condition as well, because we have a lot of immune cells in our periphery that also rely on these same receptors. And so leaving someone in sort of this compromised state isn't necessarily the same approach. So once again, we don't want to solve one problem, but then cause a different problem. And this has really sort of led to the development of these microglia replacement strategies. We heard a little bit about that from Dr. Kufel, that we've noticed within the aging brain, there are peripheral cells that can make their way into the brain, and they can take on a microglia-like identity. And so scientists and researchers have really examined this across a number of different disease models within mice. And they've gotten really, really interesting and powerful results. But as Dr. Kufel highlighted, these same studies and many, many more have also shown that these cells remain distinct. They remain different. They become microglia-like, but they don't quite make it all the way there. And what we're seeing even in some recent work, that these peripheral cells, whether they come from the bone marrow within the femur or the bone marrow within the skull, they don't even look like one another. They can look very different depending on their site of origin. And so this has been really important to understand, well, if we want to translate this to a patient, to the human condition, what's the effect that this will have within the brain as well? How important are these differences? And so there's also been some recent work within mice that have been showing that as these cells come in, they can remain pro-inflammatory, they can remain hyperphagocytic, they can induce some cognitive deficits as well. And as Dr. Kufel highlighted, within her lab, within our lab as well, independently we found that the human cells, they're much more exaggerated. And so they may not be the ideal cell, although the strategy is a very important and powerful one. And so this really brings us to what we're doing within the Blurton-Jones lab. Work that was really inspired by previous discoveries and work within the periphery, that immune cell-based strategies are becoming really powerful treatments, especially like the success we've seen with the treatment of blood cancers. There's a recent Nobel Prize given for the discovery and the use of CAR T-cell therapies, for example. And so we've been thinking that by using an immune cell from its natural brain environment, we'd be able to address many of these previous limitations by also circumventing the protective qualities of the blood-brain barrier. We're able to return those cells back to their natural environment and allow them to do their natural job of keeping the brain happy, regulating all the cells, and all the other functions that Dr. Spitali mentioned earlier. And so as the primary innate immune cell of the brain, microglia stand out as the ideal candidate for trying to develop these strategies. And so this really inspired us to ask this question. Could microglia that are derived from patient-derived induced pluripotent stem cells be used as a therapeutic strategy in the central nervous system for neurodegenerative diseases? And so this is the work that we've been really focusing on for the past 10 years or so. And so this has really allowed us, first and foremost, to develop a protocol that allows us to develop a large number of microglia from patient-derived iPSCs. We've also developed appropriate models that allow us to transplant these cells into mice. We can study the human microglia biology within these animals, not just in a dish, but in the context of all the other signaling and all the other cell types that are present. And we've also crossed these animals to other neurodegenerative models, and now we can understand how are the microglia interacting with these pathologies, and what can we do to enhance their function or rescue their function. And this has also led to the discovery of an inhibitor-resistant strategy. This is a strategy that I just want to highlight for 1 second, whereby we're able to introduce a single amino acid substitution. We're able to introduce a mutation to these cells that renders them resistant to those inhibitors I mentioned earlier that would normally deplete them, eliminate them. And I hope you can appreciate in just this image, in that first central box right there, a wild-type microglia that's exposed to these inhibitors would normally upregulate a bunch of genes, downregulate a bunch of genes, because they're slowly dying. But the cells we're able to engineer act as if that drug's not even there. And so the power of that is we can put healthy cells into the brain environment. They carry this resistance. We can administer this drug, and eventually those cells will begin to take over and replace all the surrounding microglia as well. So we're able to replace those dysfunctional microglia that are carrying those mutations and replace them with healthy, rejuvenated microglia that we're able to derive from patient samples. And so what's really cool too is that these cells will make their way down into the spinal cord as well. And we've also found that if we stop the treatment of the pharmacological, the drug, the cells will remain in these newly engrafted territories. And so this gives us a lot of hope that as microglia naturally self-renew every 4 years or so, these cells that have engrafted within their new areas will also begin to replenish themselves as well. So essentially, it's a perspective of like a living drug that's residing within the central nervous system. And so this really led us to the work that we started doing with ALSP, asking, are these cells even therapeutically potent? Can they prevent neuropathology from developing if we administer them early on? Or can they reverse pathology after it's already set in? Because in many cases, even when individuals are asymptomatic, pathology can already begin to exist within the brain. And so in order to really understand this, we did this study that I want to highlight here. And it was really focused on the observation that microglia express almost uniquely the CSF1R receptor. And in patients that carry mutations within CSF1R, individuals have lower microglia. They have an opening of that microglia niche, an emptying of the niche, which leads to the development of calcifications. It can lead to the development of these axonal spheroids that Dr. Spitali mentioned earlier, and a number of other pathologies as well. And so, this will lead eventually to cognitive and motor deficits. And working within the lab, we wanted to find a model that allows us to replicate this. Not only the opening of that microglial niche where there's less microglia present, but also the very complex pathologies that emerge due to the many, many functions that microglia play within the brain. And so we had been working with this fire model. This fire refers to an element that's responsible for turning on CSF1R within the brain. And so these mice are born grossly normal, but as they grow, a healthy mouse shown on the left would exhibit, am I able to use this? No. The healthy mice on the left, they have microglia present, but the mice that carry that fire mutation don't have microglia. And so we're able to follow these mice, we're able to watch them age. And having this empty environment, we asked ourselves, well, what happens when we transplant healthy microglia into this environment? And we saw that microglia that were derived from patient-derived iPSCs would not only engraft within the brain, but they would begin to proliferate, they would begin to migrate from the transplantation site. I just want to point out right here, those cells begin to move, and within 30 days, they're migrating throughout the brain and throughout the spinal cord. And what's really important is when we look at the genes that are now being expressed by these cells that were developed in a dish, collected, put through a needle, entered into the brain, we were surprised to see that they were happy, they were healthy. They were exhibiting gene expression patterns that we'd find within a healthy human brain environment. And so what is this doing to the pathology within these mice? Given that they lack microglia within the brain, they develop a number of these pathologies, many things that Dr. Spitali spoke about earlier, and we can see with a significant amount of age, they develop these RIS-positive calcifications, shown there in white on the bottom left. They develop astrogliosis, astrocytes. This other glial cell within the brain becomes activated, pro-inflammatory, is trying to compensate for the absence of those microglia, and a number of other things as well. But what we see is by delivering these cells early on, we're able to prevent those pathologies from forming. And so I hope you can appreciate here, we see in a mouse that didn't receive the microglia, we get those calcification aggregates that begin to form, but then those are being eliminated. Same with the reactive astrogliosis, same with the accumulation of osteopontin. Yes, for the accumulation of osteopontin as well. And so having observed this preventative strategy was very potent and very, very therapeutically relevant. We turned our attention back to that initial point that in many cases when someone would be diagnosed, this type of pathology would already be present. So rather than preventing it, are these cells strong enough, powerful enough? Do they have a therapeutic ability to reverse any of this pathology after it's already been set in within the brain? And so in a collaboration with Dr. Zolik, we were able to really explore this with the full perspective in mind. We got our hands on a patient skin sample. We isolated the cells that are derived from those skin samples. We reprogrammed them into these induced pluripotent stem cells. We used CRISPR to gene correct, to correct the mutation that that patient was carrying. And what we found is when we derived microglia from these lines, the line in purple on the right-hand side carrying the mutation was having a difficult time to proliferate, was having a difficult time to create and replenish itself. But by correcting that single change that's within that gene in green, we see that the proliferative potential was now rescued. And these cells were able to proliferate. These cells were able to not only engraft, but then take over these brains as well. So what happens when we transplant mice that are now 4 months old? These are aged mice. They already have the onset of those calcification aggregates and some of the other pathologies we spoke about earlier. And what we can see on the left-hand side is a microglial line that still carries that mutation was unable to proliferate. It was unable to deal with those red little balls that are showing, which are signs of those axonal spheroids we mentioned earlier. But the CRISPR-corrected line not only proliferated, not only engrafted and filled the empty niche like we saw earlier, but they began to eliminate that pathology that was there as well. And so what we can see on these graphs on the right-hand side, the first bar is just a healthy mouse, has microglia from birth, and what we see is that the pathology is not there. In blue, light blue, we see this was the mouse at 4 months old. At the time of the treatment, many of these pathologies are already aggregating and collecting within the brains. And by transplanting in purple the patient line that didn't have the ability to replenish the brain, those pathologies in some cases were even worse. But when we were able to correct that mutation, when we were able to deliver healthy microglia to the brain, that those levels began to retreat. That pathology was being eliminated from within that environment. And so this has given us a lot of hope, not only to use microglia as a therapeutic cell type in order to treat the absence of microglia, but our ability to genetically engineer them and to harness the stem cell state of these cells has allowed us to really ask sort of a follow-up question of, well, how else can these cells be engineered in order to treat neurodegenerative disease? And so I just very quickly want to highlight a follow-up study that can really show the potential of these cells. What we see here in this animal model of Alzheimer's, as it develops amyloid plaques, which is the first stage within the Alzheimer's disease, microglia that specifically come into contact with these plaques induce very unique expression patterns. We see the CD9 gene in red turning on only in those green cells that are next to the white dots, which are the amyloid plaques. And we see this happening throughout the brain of these mice in all the different areas that we examine. And so this has really presented us with the opportunity to ask, could we use this as a genetic switch to induce expression of some type of therapeutic payload, some type of therapeutic peptide at the sites of these aggregates, at the sites of injury, specifically in response to these depositions or this neurodegenerative disease. And that's what we're able to see here. In green, these are these engineered cells that are delivering this amyloid protease. This is an enzyme that chops up amyloid. And what we see is only those microglia that are coming into contact with that blue amyloid aggregate are inducing expression of that payload, that neprilysin, specifically in response and at the sites of the pathologies. So not only are we able to deliver these cells to the brain, but they can be engineered to essentially assist their natural function of dealing with these depositions, these aggregates, and these debris that can accumulate over time. And of course, we wanted to make sure we understood, could a few cells offer a brain-wide efficacy because it is a secreted payload, or do we need widespread engraftment? And without going too much into the weeds on this, what we were really excited to see and we were able to publish was that these cells, although small and mighty, from a single engraftment within the brain, they were able to reduce brain-wide levels of amyloid, they were able to reduce brain-wide levels of inflammation, they were also able to preserve brain-wide levels of these synaptic proteins as well that are really important to how neurons communicate and talk with one another. And so we in the Blurton-Jones lab are continuing to investigate this therapeutic potential. We're interested in other neurodegenerative diseases as well. As we mentioned, these cells are implicated in frontal temporal dementia, in other leukodystrophies as well. And so I want to take a second to thank everyone in our lab who's been instrumental in doing this work. I also want to thank our collaborators too. And of course, with the future in mind, our youngest new scientist within the lab, who's also given us a little bit of hope too to keep working hard on these things. And so thank you everyone. If you have any questions, I'll be happy to take them now. Thank you.
Very enthusiastic talk.
Thank you.
Very good. So we listening to your talk, it's definitely a wonderful strategy to have a replacement of microglia. Now, but how to deliver this microglia in human? There's many studies in our diseases that the ASO, for example, are delivered and or antibodies, and in my career, those trials are more and more aggressive. They started with pills, then they moved to IV infusions. Now we have PSP trial with intrathecal injections, and there are other trials for Parkinson's disease with intraparenchymal injections or intraventricular injections. So in your opinion, based on the mice experiments, are there any studies they study the delivery to the brain? Because in your experiments, you put that microglia directly to the brain,
Correct.
right? In mice, you can do it, but each time you do it, you damage the brain. So are you developing any studies that perhaps you try to do these experiments in less invasive way.
Yes.
For example, intrathecal delivery, or maybe IV, maybe intranasal delivery, right?
Yes.
Or some other type of delivery of microglia when we have that will be less invasive and it will be easier and less risk factors, no hemorrhage, no infection. You know, those are serious side effects that potentially be, even the craniotomy is a risk factor by itself in patients. So what would be your take on this question?
I really appreciate that question. And it's really important that we in the lab always keep the patient experience in mind, right? We want to come up with an idea and approach that can translate to the real world. And so that is something that we are very sensitive to. But we're working sort of in reverse to how those other drugs have worked. So we, of course, the work that I've shown you so far was done by direct injection into the brain. That's correct. And that's a similar strategy that's being done in patients for a number of other diseases too. But in order to investigate sort of less invasive strategies as well, we've published recently that delivering microglia through the cerebral spinal fluid, we're able to essentially seed the brain, and those cells can make their way into the brain tissue as well, eliminating the need to cut open the skull, to damage the dura underneath as well. So there are less invasive strategies. And there are groups that are proposing other peripheral delivery strategies as well. However, the one thing that I want to highlight in many of those claims is they are using those microglia-like cells, which originally come from the periphery. And so they have, you know, more of a tolerance to that environment. So we do have ongoing studies looking at peripheral delivery of iPSC-derived microglia. We have, unfortunately, I can't share in much detail, but we have some really exciting opportunities there. And we really hope that eventually we can get a brain-targeted approach. But of course, being able to administer these cells within the CSF alone by cisterna magna or intrathecal within mice has already shown that there are the ability to do so. And so we really want to capitalize on that and figure out how else we can reduce the patient burden. You know, if it's a lumbar puncture, which is an outpatient procedure, that would be remarkable if that's the case. Yes.
I have a next question. What will be the dose and how many times you need to do that in order to be successful? Is it one-time delivery to
Sure.
the brain will be sufficient? And you think that if you do that intra-brain or intraventricular, let's say, perhaps less invasive, do that microglia cross the other hemisphere through corpus callosum? Is it possible, or are they migrating across the brain?
Yes.
In mice?
Yes. Microglia are very, very dynamic cells. And one of their most unique properties that I find very interesting is their ability to travel through solid tissue, which is very unique to any other cell type within the body. And so if we were to deposit cells on one side of a brain, and especially if there's an emptying of the niche, those cells can travel through not only the brain, but the spinal cord as well, and they can make their way down as well. And there's some initial evidence we find within the lab that even cells that were direct deposited within the brain eventually can hitch the cerebral spinal fluid flow and can proceed to seed other parts of the brain too, which is really exciting as well. And so we have noticed in a microglia depleted environment where there's no microglia, the entire CNS is engrafted. Now, in an environment where there are microglia present, they have a really interesting ability to tile themselves and to maintain their numbers, because too much microglia is also a bad thing, right? Too much is bad and too little is not good as well. And so those cells will begin to fill in those empty spaces. But as the microglia begin to self-renew, they have the ability to travel along the white matter. And we find that transplantation, even into the forebrain, will eventually give rise to these human-derived microglia within the rest of the brain as well. Although not in significant numbers, but they can begin to make that journey. That's why something like that inhibitor-resistant approach, it allows us to control the environment. We can regulate the area that, or the regions that we want to engraft, or over a long-term treatment, potentially replace all of the surrounding cells as well. And so in terms of dosage, the mouse work we have so far would suggest that over 14 months, these cells remain, they don't retract from their engrafted areas, they're able to persist. They seem to exhibit the same, sort of lived cycle that microglia naturally do within the brain, where they begin to self-renew. They're not retreating from their territories. And so we would hope to see that within larger animal models and eventually within patients, that they would also be a lived drug, where we can deliver these cells, they'll engraft, whether it's a single dose or a multi-dose, those things need to be discovered or worked on a little bit more. But we would hope that it'd be a long-term therapeutic strategy that would be able to accompany someone throughout their life as well. of how to get this into the clinic? Oh, I'm going to point your attention to Dr. Rob Spitali, who will speak a little bit more about those, but yes.
I've got a quick question. It's about the dosage and how, so when we culture microglia in a dish from iPSCs, they're terminally differentiated. They won't divide. So you just said that when you put them into the mouse brain, that those cells are allowed to, you said self-renew. Does that mean that it can divide? Does that mean that they gain the ability back to divide and repopulate the brain, so they don't just engraft, but they actually divide?
Yeah, great question. So whenever we derive microglia with our protocol, we initially go through this progenitor state, which is highly proliferative, and those cells are dividing and giving rise to tons of microglia. But as you mentioned, as these microglia mature, eventually in the dish, they lose the ability to keep dividing. Those cells at the end of that stage, if you put them into an open environment, they regain the ability to now divide and to take over that empty niche as well. And so we can use techniques within the lab to really look at that using Ki-67 staining, for example. We can look at cells throughout different time courses in mice that were transplanted at 1 week, 10 weeks, and so on and so forth. And we see that those cells are taking over. As they're proliferating, they exhibit that profile you see within a cell that's dividing. But as they engraft within that new territory, they revert back to that healthy, happy, homeostatic profile. And so we've seen that even by engrafting the entire CNS of these mice, these cells will then revert back to this happy, healthy, homeostatic profile as well.
So you think that if you, for example, if you test that by maybe partially depleting your transplanted mice, using the usual chemo treatment that they give, do you think these will be able to repopulate the whole brain again?
I don't know specifically about using like busulfane or some of the conditioning drugs, but using the CSF1R inhibitors, yes. So we've shown that within these mice that we're able to engraft the cells, we can then reduce their numbers, and if we take the drug away, they bounce back, they then regrow. So they're able to, they exhibit the self-renewing ability that microglia naturally do within our lives, within our brains, yes. What?
Is it possible to do these experiments in primates?
It would be possible, and that's something we would love to do. Yes.
Okay.
Thank you so much, everyone. I really appreciate it. Thank you.